Welcome back to Science of Medicine. Today I want to talk about the electrical activity of the heart and specifically how we can, as engineers, actually tell something about the heart and its health from EKGs. Now remember that certain cells in the body are polarizable. They can have a potential difference between the inside and the outside of a phospholipid bilayer. And that separation of ions is something that allows for those cells to be excited. Things such as nerve cells or muscle cells have transmembrane ion channels that allow those ions to pass once there is an appropriate stimulus. We've talked about this for skeletal muscle when acetylcholine binds at the synaptic junction on the muscle side, opening up sodium channels and allowing that to initiate depolarization of the muscle. Now, which direction the ions flow spontaneously depends upon the various concentrations of those ions. So I show in this table typical ions that are present in reasonable concentrations. The blue represent the cations and the red the anions. Now, for the most part, sodium is our predominant cation in the extracellular fluid. And its concentration is around 140 millimolar outside as opposed to inside 14 millimolar. Therefore, if I open a sodium ion channel, the spontaneous transport will go from extracellular to intracellular. This is why when I depolarize a muscle cell, sodium rushes in when I open those sodium channels. Potassium is the reverse. Potassium has a much higher concentration on the inside of the cell than on the outside, about 140 millimolar versus about 4 millimolar extracellularly. And so if I open a potassium channel, then potassium will flow spontaneously from inside the cell to outside the cell. Those spontaneous processes are driven by that concentration gradient. But in order to get those concentration gradients, I have to have active transport that is an energy requiring process. And that can be through either a symporter, where I transport two substances in the same direction, or an antiporter, where I actually have one ion transferred in one direction, another ion or another substance transferred in the opposite direction. Now, if I look at various types of examples of that, an antiport that moves ions from one direction to the other. In the loop of Henle, there are sodium-potassium chloride transporters that serve this purpose. The sodium-potassium ATPase that is present on the muscle cells is another example of that. Symporters, I have a symporter in my gastrointestinal tract that moves a sodium ion plus a glucose into the cells. And this is the basis for Gatorade and other oral rehydration solutions. Now I can actually model and calculate the concentration difference across a cell that is excitable and the potential difference that results as a result of that. The Goldman equation, which is basically the Nernst equation in this particular case, is a description of that. You may know from general chemistry that the Nernst equation looks at the potential difference across 2 points that is related to various variables, things such as concentrations, technically activities, and then also a term which depends upon Faraday's constant, number of electrons transferred, the ideal gas law constant, and the temperature. So if I look at this adapted to mammalian cells in general, what I find is potential difference across a cell membrane is given by a term, this minus 61.5 millivolts, is that 2.303, which is a conversion between log base E and log base 10. RT, R being the ideal gas law constant, T is the temperature. In this case, it's 310 Kelvin or 37 degrees Celsius. Over NF, N being the number of electrons, in this case for the cationic species +1 or anionic species -1, and F being the Faraday. And the logarithm of various concentrations with their permeabilities. Now, permeabilities are simply sort of a fudge factor to account for differences in measured cell potentials. For example, it may not be completely dependent upon potassium, it may be partially dependent upon potassium as well as concentration. So the permeability of that ion times its concentration on the inside versus the outside for the cations. The chloride, which does not play a significant role for most human cell lines, is switched, it's outside over inside, so that I can actually have the negative 61.5 millivolts. You may remember that I could describe a log of something as sort of A log of B, or I could say minus the log of minus A times the log of 1 over B, and that's why this chloride is flipped over on this. And these are additive. So if I look simply at a particular ion, let's say potassium, then I call that permeability 1, and the permeabilities of the others 0, and they drop out because they're additive. So if I look at my typical cells, where I have 140 millimolar inside the cell of potassium and outside about 4, 142 millimolar outside of a cell for sodium, as opposed to 14 on the inside, I can pump using this sodium potassium ATP pump to make this in balance. And in fact, I pump out 3 sodiums for every 2 potassiums in. And I also have some leakage channels that are also allowing for this disequilibrium position to be maintained. So just as an example, if I ask you to calculate the resting membrane potential of a myocyte, if the potassium is the main determinant, and give you the inside the cell potassium concentration of 140 millimolar and the outside 4 millimolar, you should be able to do that using the Goldman equation. Remember, in the Goldman equation now, my permeabilities of sodium and chloride are going to be 0, and so these are going to drop out. So I'll give you a second to try and calculate that on a calculator. So I hope that you recognize that this Goldman equation simplifies quite a bit. I still have my minus 61.5 millivolts and the log now of the inside over outside potassium concentration, because the sodium and the chloride have dropped off, because the potassium is the main determinant. And when I do that calculation, I get about a negative 95 millivolts. This is not far off from the resting membrane potential of many of the excitable cells in the human body. And again, it's not exactly matching because that permeability is not 1 in this simplistic example. Now remember, we talked briefly about action potentials before, and so if I measure this voltage, in this case minus 70 for some excitable cell, that if I have some sort of stimulus, if I get above a certain threshold, I will allow for depolarization of that cell. For example, if this is an example of a muscle cell and I have a nerve that is impinging upon that neuromuscular junction, releasing acetylcholine, which is binding with the muscle's acetylcholine receptor and allowing for sodium to be, sodium ion channel to be opened, then the sodium will be moving from the outside of the cell to the inside, and this will cause a change in potential to a more positive value. Now there are voltage-gated potassium channels that will also be activated during that depolarization. And when that happens, potassium will move from the inside to the outside, as well as the sodium channel then closing after a short period of time. And this is why the potential peaks and then goes down more negative. I'll overshoot a little bit and have a small refractory period when I can't excite this cell, and then maintain or then reestablish that resting state as I'm using this sodium potassium ATPases to go back to that that original state. So let me remind you of some things as this applies to the heart. Remember that the cardiac myocytes are connected via intercalated disc. Those are specialized gap junctions between the myocytes that allow for very fast diffusion of ions through them to allow for very fast transmission of those depolarization signals. And remember further that in the heart, I have in the SA node of the right atrium a set of intrinsic pacemaker cells that depolarize without acetylcholine. Now I can modulate them, I can have epinephrine from the sympathetic nervous system, make them go faster, or also parasympathetic to slow the heart rate to be able to do this. But they are beating at an intrinsic rate of 60 to 100 beats per minute. And if you look at their depolarization, it's a longer period, not just a few milliseconds that actually was present on the last slide. And that's because there are several channels. There are two types of sodium channels, a fast and a slow sodium channel. And then there's also a calcium-sodium channel. And that leads to a lengthening of the plateau so that I get polarization and depolarization over a longer period. Now not something like a few milliseconds, but instead more than 100 milliseconds, so that I get transmission of the electrical signal in the heart going in one direction and not going backwards. So if I actually were to look at this, those sodium channels that are actually causing that potential to be over about 100, 150 milliseconds are essential so that I get that unidirectional SA node going through the atria and going to the AV node propagation. So let's talk a little bit about this. If you remember when we were talking about cardiac electrophysiology, we mentioned that the pacemaker cells are located in the right atrium, the SA node, and they will beat 60 to 100 times per minute. Now, once I've actually had that initiated, those signals will propagate through the atria. There's branches that go off into the left atrium, and they'll meet up at slightly different times at the AV node. And there's a brief pause at that AV node. And that's important so that all the signals propagate forward down the bundle of His at the same rate, at the same time, excuse me. So once I actually have that bundle of His propagation, I'll point out that I have sort of a branch that goes to the left side and a branch that goes to the right side of the heart. And then those branches then go to the apex of both the left and the right ventricles. Now I do have backup pacemakers that are in the bundle of His that will initiate contraction if the signal does not make it through the AV node, and I also have backup pacemakers that are in the ventricles if I would not get a signal from the bundle of His. So it turns out that you can measure this, and this was measured more than 100 years ago by a biomedical engineer, before there was biomedical engineering, who placed electrodes on the surface of the skin. And it turns out that we use this clinically many, many times. And it's called an EKG or sometimes an ECG. And so what we note is we can put surface electrodes in various places around the heart. And there are so-called precordial leads which go around the heart, as well as limb leads which are on the arms and the legs. And I can take the difference in potential between those two, or between any two, and get a series of snapshots of the electrical activity of the heart. Now, I place 6 precordial leads, 6 leads around the heart, and then the 4 limbs, so I have a total of 10. But on an EKG, I'll actually have 12 differences in potential. And they'll give me indications or snapshots of various physiologic areas of the heart, the right side, the left side, as well as other areas of the heart that are important for me to determine where there might be damage or where there might be stress on the heart. So let's talk a little bit about what that looks like. So what that looks like is I get this repeated kind of pattern, and I call these waves going up or down by letters in the alphabet. and I start with P and go out to T. So the first change or deflection or peak that I will see is what we call a P wave. Now technically I get a little bit of depolarization of the right atrium before the left atrium. In many cases you can't see that, it's a broad wave. Sometimes, especially if you have one atrium which is bigger than the other, you can see those two separated somewhat. But that first P wave represents the depolarization of the atria. And then I'll have a Q wave, which is a downward deflection, an R wave going up, and an S wave going down. And that represents actually several things. It is the repolarization of the atria as well as the depolarization of the ventricles. Now, the time that it takes to get to that QRS wave is called the PR segment. Remember, I said that there was just this little pause, if you will, and that's indicative of this. That's indicative of this pause. Now, once I actually have gone through and depolarized my ventricles, repolarized my atria, I'll then have to depolarize my ventricles, and that's a T wave. Now, there are other waves that can actually change. For example, under certain conditions, particularly with potassium, I can have another wave, and that's just called the U wave. We won't talk about that beyond this in this class. There are several segments that you may have heard of before. So first off, the ST segment, that is the segment between the S wave, which is downward, and the T wave, is something that is looked at to look at myocardial damage or ischemia or difficulty with blood flowing. You may have heard of an ST elevation myocardial infarction, where I've actually clotted off a blood vessel that is on the heart, causing damage, and that will lead to this potential moving upwards. Notice that I can have time, since this x-axis here is my time, my y-axis is my potential, and so I can have prolongation of different segments caused by different reasons. We'll talk a little bit about some of those. Now, there are other rhythms other than sinus rhythms. I can have other atrial rhythms, and these signals don't come from the SA node, they come from another location in the atrium. And I'll talk about two of the several atrial rhythms, atrial fibrillation and atrial flutter. There are some times where the signal does not originate in the SA node or the atria. For example, junctional rhythms, where the signal is coming from the AV node, or ventricular rhythms, where the signal is coming from backup pacemakers in the ventricles. I'll also touch on conduction abnormalities. These are electrical conduction abnormalities, such as heart blocks or bundle branch blocks, and then mention just a little bit that sometimes there are other pathways other than what we've just discussed that can cause arrhythmias. Now, in the old days for EKG, we used to actually record them on a strip chart recorder. That's done electronically now, digitally, but many times it is plotted on this. And so just to sort of give you a way of looking at these plots, I want to show you that on these grid paper that you'll see it printed on, that the y-axis refers to a potential amplitude. And there is a setting so that it says that it's 1 millivolt for every 10 millimeters high of these boxes. And likewise, if I look at one of the large boxes on the x-axis, which is time, that's the same as 200 milliseconds. If I divvy this up, I can count from my peaks. The first big box would be a rate of 300, next 150, next 100, next 75, 60, 50, 43, 37, so on, just basically looking at that 200 milliseconds. So let's talk about some of these. I mentioned that one of the rhythms that we have are sinus rhythms. Sinus rhythms always originate in the SA node, and there is a P followed by a QRS. So I want you to look at this top EKG, which is showing 12 leads, 1, 2, 3, and then we call them a little bit differently in terms of this. There are 3 more here, and then these are the precordial leads, V1 through V6. And you'll notice that the potential is along the y-axis. So here is an R wave, here is an R wave. And the time is along the x-axis. So I'm going to go for any one of these. And if you look at this, you can see that there's a P wave followed by a QRS, and a P wave followed by a QRS. This is a sinus rhythm. And since the rate, which you can either read off the EKG or you can calculate it based on the distance between the R and the next R wave, would be 62. And that would put it between 60 and 100, which would be in the normal sinus rhythm. And contrast that with the EKG that's shown down below. Now in this case, I have a tracing down here, which is just simply lead number 2, which has been expanded out across this time. And if you look at this, I still have a PQRS2, PQRS, and I have T, PQRS. So every P has a QRS wave. Every P has a QRS. So this is a sinus rhythm, but now the rate is 140. It's greater than 100, so it is a sinus tachycardia. Now, one of the ways that this is accomplished is by having the sympathetic nervous system act upon the SA node to speed this up. For example, let's say you've been running and you have adrenaline pumping through your system, and that adrenaline then is going to actually impact your heart rate so that you have increased cardiac output. So that's a sinus tachycardia. Similarly, if I show my EKG here that I had that was normal sinus and contrast it with this tracing, which is a sinus bradycardia, I have a PQRS, PQRS. This is a sinus rhythm, but now since the rate is less than 60, it's 50 in this case, it actually is a bradycardia. And this can be a result of the parasympathetic nervous system acting on the SA node to slow the heart rate down. Now I can't slow it any slower than when the backup pacemaker is in the bundle of His fire, because if a signal does not come through fast enough, come through the AB node, then those backup pacemakers will fire. So again, sinus rhythms always originate in the SA node and have a PQRS. There are other places that you can have signals originate in the atria, and they're not in the SA node. These are not sinus rhythm. These are other atrial rhythms. 2 of the more famous ones are atrial fibrillation and atrial flutter. So if you look at this strip right here of sinus rhythm, PQRS, 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 this is a sinus rhythm, and compare it with this irregular. I have this sort of jagged baseline, and it's not a clear P wave for any one. This is because there are multiple foci of where those signals are originating, not just in the SA node. And this is called atrial fibrillation. You can see that the spacing between the RR intervals is constant in this sinus rhythm, but irregular in this atrial fibrillation. You also see a case of atrial flutter, which is what we call a sawtooth pattern, which is here. And this originates mostly around the tricuspid valve, and in this case, the signal is not conducted every single time through the AV node. You have conduction after every so many sort of signals that go through. So there's normally a block of 2 to 1 or 3 to 1. Those are atrial rhythms that are not sinus. So another possibility is a junctional rhythm. A junctional rhythm is where the signal is originating in the AV node, so between the atria and the ventricles. And in this case, if you look at this strip down here, which is 2, you won't see any P waves. There's no P waves. There's a QRS and a T, QRS and a T, QRS and a T. This is what's called a junctional rhythm. There actually is a P wave. It's buried in that QRS wave. Sometimes for junctional rhythms, You'll also see an upside down P wave where you have retrograde transmission of the signal through the AV node backwards into the atria. This is not normal. This is an abnormality. This is a junctional tachycardia. The rate here is actually fast. And the key thing here is that you see in none of these leads any P waves, but it's a very regular rate that is going on. And this is a junctional rhythm. It's also possible for you to have a ventricular rhythm where you use those backup pacemakers. And in this case, there's also no P waves, but the rate here is exceptionally slow and the QRS complex is very wide. And there are certain criteria for this, but it's a widened QRS. And in this case, the rate is 25, which syncs with that 20 to 40 for the backup pacemakers. Now you can have ventricular rhythms that are very fast using other pathways, and we'll touch on that just a little bit. But these are ventricular rhythms. No P waves, widened QRS. There are also things called heart block. Now a heart block comes in different forms. The first is where I actually have a slowing of the signal through the AV node, which leads to a PR elongation. So in this case, the PR interval here is bigger than one of these big boxes, which is 200 milliseconds. So when that happens, and that's the only thing that happens, it is called a first-degree heart block. There's a possibility of having a second degree, but it comes in two different types. A Mobitz type 1 or a type 1 block, I'll have the PR interval get longer and longer, and then I will drop a beat. So second degree heart block, I drop a beat. But the PR interval in type 1 gets longer, but in type 2, it's the same. It stays constant, and then I just drop a beat. So here's my P wave, and I don't have a QRS. Upward here, I had a PR, but I don't have anything. It drops and then it goes back. This is a type 2 block, or a second degree block, I'm sorry, in type 1 or type 2. Type 1, elongated PR followed by a drop beat. Type 2, constant PR with a drop beat. And then the last type of heart block is a third degree. Now a third degree heart block, the atria are doing their thing and the ventricles are also doing their thing, but they're not electrically connected. There's no correlation between the P waves and the QRS complexes. So I'm going to look at our rhythm strip down here, which is lead 2. And if you can see, I have a P wave here, got P waves, got P waves, got P waves, P waves. But they're not really doing anything related to the QRS. Here's a QRS, 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 QRS. This is a third degree heart block. And it's important to recognize these because this requires that you take an intervention, in this case a pacemaker. And it's true also for type 2 second degree heart blocks, because those will many times degrade into a third degree heart block. Now I mentioned before that when you get to the AV node and go forward, I have a branch of the bundle of His that goes to the left ventricle and a branch that goes to the right. And I can have blocks in just one, not both, of those pathways. And that leads to a type of heart block, which is either on the left side, which I call a left bundle, or the right side. Now it turns out that if I want to see these, I'm going to look in two leads, the precordial lead V1 and the precordial lead V6. And I'll have a certain pattern, okay? I'll have the pattern that I'm looking for in terms of this. In this case, for a left bundle branch, you'll notice that there's a widening of the QRS, and this is always the case for bundle branch blocks. But the shape is what I would want you to note, and I'll show you when we talk about the right bundle branch that there's a difference. you can see in V1 that I get this RS kind of wave. And if I look at the right bundle branch block, where I've done this, I still get a widening of the QRS, but now in that V1, I get a different view. I get sort of this RS R prime. So the R goes back up again before I go to the T. So those are bundle branch blocks, and they're also indicative of conduction abnormalities. Now I'm going to touch on an abnormality that occurs that I'm not even going to begin to talk much about, but those are called accessory pathways, and there are a number of these. Accessory pathways are where you don't go through the AV node, but you have some connection electrically between the atria and the ventricle. And these can lead to very bad arrhythmias that can even be lethal. So there's one type which would be typical in young college age students that's called AV nodal reentrant tachycardia, where there's a pathway between the atria and the ventricles that can be shown. There are others that are very pertinent that can cause tachycardias, such as Wolff-Parkinson-White, and those are well beyond the description in this course. But I wanted you to know that there are other abnormal pathways that can exist. So I hope that this has given you a little bit of an introduction to how the heart is actually conducting itself in terms of electrically, and I'll see you back when we talk about medical imaging.